Okay, now we're going to talk about the erythrocyte. Remember, erythrocyte is the same thing as the red blood cell. They all have the same form and function. The function is basically to transport gases, mainly oxygen. The form is called biconcave because if you're looking here, we have one concavity here, right? This part is concave and this part is concave. The red blood cells, one very important characteristic that they have is that they do not have nucleus. No nucleus, no cell organelles, in particular, no mitochondria. What does it have instead? What does it have inside here that allows it to transport gases? Well, inside there, what it's going to have is going to be hemoglobin. If you scroll down, so again, job of the red blood cell is to transport gases. That's not the only job. It also helps us regulate the pH. That regulation of pH is controlled by this chemical reaction that you see here. Carbon dioxide plus water is going to give you carbonic acid. The carbonic acid is going to dissociate into hydrogen plus bicarbonate. So right here again, we have hemoglobin. Now, what's the composition of hemoglobin? Hemoglobin is made of heme and globin. This is the arrangement of the hemoglobin, where the heme is this red part right here, and the globin is going to be this brown or blue chains that you're going to see right here. So how many globin chains you have? 4. 2 blue ones, also called alpha, and 2 brown ones, also called beta. In addition to that, each globin must have a heme, so that's why you have 1, 2, 3, and 4 hemes. So this is one hemoglobin, another one, and another one. Hemes here is mainly red, but right here in the middle, what you're going to have is iron. Hemoglobin will allow the red blood cell to transport gases. It says in here quantities of erythropoietin and hemoglobin. What is the concentration of hemoglobin? Approximately 12 to 16 grams per deciliter. The lower the value usually belongs to females, higher the value it belongs to males. The erythrocyte life history, basically where it's created, for how long it's going to live, and where it dies. The red blood cells, or erythrocyte production, is going to happen in the bone marrow by a process called erythropoiesis. Erythropoiesis is a process by which stem cells, as you can see here in this graph, stem cell, it has the ability to produce red blood cells. You're going to see other graphs where the same stem cell is going to give you white blood cells, and the same stem cell is going to give you platelets, because the stem cell has the ability to produce multiple different lines of cells. What hormone stimulates the production of red blood cells? This hormone is called erythropoietin. The erythropoietin is a hormone produced by your kidneys. When we lose red blood cells, the kidneys are going to get this information, and as a consequence of that, it's going to produce erythropoietin. Erythropoietin will travel to your bone. The stem cell is going to get stimulated, and you're going to produce more red blood cells. Now, once you produce a red blood cell, for how long does it live? 120 days. After that, they become old, and usually they get trapped and destroyed in the spleen. What happens when you destroy the red blood cell? Then obviously what you're going to have is what is left inside the red blood cell, which is going to be the hemoglobin. The process of decomposing the hemoglobin is going to start at the level of your spleen, where you have macrophages, right? These specialized cells that are going to eat things. So the macrophages in your spleen are going to eat this hemoglobin, and they're going to start decomposing the hemoglobin. Globin is a protein. So then when the macrophage gets the hemoglobin, it's going to break it into pieces of amino acids. The macrophage is going to get the heme, and it's going to destroy the heme first. It's going to transform that into biliverdin, and then it's going to transform that into bilirubin. Heme, we said that in the middle is going to have iron. So the iron is going to be also separated and is going to be stored. So where do we produce red blood cells? In the bone. After they are produced, for how long do they live? 120 days. What happens after 120 days? They get trapped in the spleen. What happens in there? The macrophages in the spleen are going to eat the hemoglobin by a process called phagocytosis. Here's the hemoglobin that was phagocytized by the macrophage. What are the components of the hemoglobin? Heme and globin. So the first step in destroying this inside the macrophage is going to be to separate the heme and the globin. We said the globin is just a protein. So then that part is going to get destroyed into amino acids. Here you go. Now we said the heme has 2 parts. So the heme is going to get separated into iron, which is going to get stored in the liver for reuse if it's necessary. And the other part, the heme, is going to get transformed into biliverdin, bilirubin, bile, and then it is going to go to the feces. Now we're going to start talking about blood types. Now, if you look at the first concept that it says in here, This is a compatibility issue between plasma proteins and erythrocytes. So when we talk about plasma proteins, we are talking about the antibodies, right here, antibodies. And when we talk about the red blood cells, you're talking about the antigens. That's why these two items are being highlighted in here. So there's going to be a good relationship between the antigens and the antibodies in order to make this interaction work. Otherwise, there's going to be a problem. Now, what type of interaction is this? Well, this type of interaction is going to be called the antigen-antibody complex. If this interaction is not the most appropriate, is not correct, then this interaction is going to get bad, and you're going to get a problem called agglutination. And obviously, the person getting the blood is going to start suffering. Okay, so let's just scroll down. Right here, we have medical history. Obviously, we are not going to talk about that. Right here, we have a table showing us the ABO blood group.
Blood type AB has antigens AB, but no antibodies. Blood type B has antigens B, but antibodies anti-A. Blood type A has antigens A and antibodies anti-B. And blood type O has no antigens, but has antibodies anti-A and anti-B.
Let's continue with the ABO group. Still in here, we already look at all this information. And the next topic is going to be the RH group.
The RH group includes antigens C, D, and E. Antigen D is the most common, so the person is considered RH positive if the person has the antigen D, and RH negative if the person does not have it.
There's conditions related to the RH group, erythroblastosis fetalis, or hemolytic disease of the newborn, which is something that happens with the RH groups. So let's continue other blood groups. Besides the ABO and RH groups, there are some other ones that you can see: Kell, Kidd, Duffy, and these also may create, as you can see here, hemolytic disease of the newborn, unless that you check and make sure that you verify that the parents are not at risk for hemolytic disease of the newborn if they have a baby. Because again, if you get the wrong combination, then obviously the blood is going to get destroyed. So if we scroll up, we get right here to this figure where you can see that when you have the wrong compatibility, the blood gets destroyed like that. In this case, the compatibility was correct, so you can see that nothing actually happens. Obviously, in this case, you see that something was destroyed, and you can clearly see that this is type AB. In this case, nothing happens, so it's going to be type O.